cholesterol was first described by François Potier de la Salle, who found it and isolated it. This is a guy, you can see low resolution back in 1767. Just joking. He isolated cholesterol from this. Any of you have an idea what that is? It's about the size of a baseball. This came from a cow, to be precise. You could find something like this in humans, but it would be smaller. It's almost pure cholesterol. No? No idea? This is a gallstone. So there are different kinds of gallstones, but the predominant form of gallstones is a cholesterol gallstone, and this is really crystalline cholesterol. The molecule then later was by another Frenchman actually named cholesterol, which was derived from two Greek words, chole, which means bile, which is your connection to the gallstone, and then steros, solid, because it was in the gallstones, and then the ending OL, which is the convention for alcohol, because he knew it was an alcohol. The molecule is really coveted when it comes to getting Nobel Prizes. If you want to get a Nobel Prize, I highly recommend anything with cholesterol. No molecule got more shout-outs. I think 30 Nobel Prizes. Of course, one famous one for this man here. Do you guys know who that is? No? Konrad Bloch. He's the guy who actually deciphered the many, many steps in the synthesis of cholesterol. Then, of course, these two here. Any idea who that is? Brown and Goldstein, that's right. So they got the Nobel Prize for the discovery of the LDL receptor. And of course, today's topic of discussing cholesterol is intimately linked to the discussions that you already had at the very outset of the semester about lipoprotein metabolism, as the principal function of the lipoproteins is, of course, to transfer cholesterol both towards cells as back from cells and out of the body through reverse cholesterol transfer. And you've already learned from Dr. Krauss that a disbalance in your lipoprotein profile and an increase in the circulating cholesterol levels, particularly in LDL, leads to what disease? Atherosclerotic disease, right? But cholesterol is also incredibly important to be regulated on the cellular level, and each cell has to make these decisions for herself. And that is because without cholesterol, membranes would be way too rigid, and you actually would develop holes in the membrane. On the other hand side, if you have too much cholesterol, you will kill the cell due to oxidative damage and overly fluid membranes that get too much curvature. And the cell needs to strike, just like we discussed with iron, a delicate balance of having just enough. You have to be in the Goldilocks zone. Not surprisingly enough, for such an important thing, there are two pathways that actually regulate the abundance of cholesterol within a cell. The first one involves a transcription factor, and the second one involves a biosynthetic enzyme. We shall discuss both. Cholesterol can be derived from, of course, the diet, and I briefly should backpedal here. When the cell senses cholesterol, there can be two principal sources of the cholesterol. One can be from the diet, and of course the transport and absorption of cholesterol in the intestine can be regulated. But you also, unlike iron, where we also had regulation of absorption during the last lecture, what we discussed, remember that? Unlike iron, you can of course synthesize cholesterol de novo. In each cell has the biochemical machinery to make cholesterol. And indeed, you can make quite a bit of cholesterol if you eat a diet that is pretty low in cholesterol. You can make around 800 milligrams of cholesterol a day. Just synthesize it yourself. This rate of cholesterol synthesis on the cellular level is extremely tightly regulated, as I just explained, because you need it, but at higher concentrations, it can easily become toxic. So we have two feedback mechanisms. The first one I want to talk about centers around a transcription factor called SREBP. This stands for Sterol Regulatory Element Binding Protein, SREBP. It's called SREBP because it binds to elements in the DNA that were termed sterol regulatory elements, because when you study just on a gene expression level, what gets expressed when you have either too much or too little cholesterol, what gets regulated, people found that these genes share a common lead sequence here, and the promoters that SIEBP can bind to. Now the protein then was subsequently isolated after people knew what DNA sequences it bound to, and this is the organization of this transcription factor. The first part holds very little surprises. So this is a basic helix, loop helix, zipper transcription factor, meaning that it has long alpha helices that form a dimer and can bind to DNA, and at its end here's an activation domain that can recruit co-activators. So far, so boring, right? But then when people looked at the sequence of this gene, it didn't just stop here. The next thing that comes are two transmembrane domains. And now that was a huge surprise for the people that cloned the gene. They thought they made a huge mistake. Maybe this isn't the right gene. I mean, how can a transcription factor have transmembrane domains, right? They will put it inside of membranes. And then there was this regulatory carboxy terminal domain that we wanted to discuss. But the transcriptional factor with transmembrane domains was in fact a true surprise, and it led to the unraveling of a really elegant regulation mechanism of nutrient sensing that intimately depends on the presence of these two transmembrane domains. So what happens when a cell synthesizes the SIEBP, this transcription factor protein, is that because of its two transmembrane domains, it is co-translationally inserted into the membrane of the endoplasmatic reticulum, the ER. Now, of course, while it's in the ER, it cannot work as a transcription factor. That goes without saying. Once it's synthesized and inserted into the ER, SIEBP always binds to a partner called SCAP. SCAP stands for SIEBP cleavage activating protein, and I'll explain how that name came to be in just a moment. This is a mandatory and obligatory binding that automatically happens. Now keep in mind, this is a transcription factor that gets activated in the absence of cholesterol and enhances the synthesis of proteins that are involved in both the uptake of cholesterol as well as the synthesis of cholesterol. So it somehow needs to be able to sense cholesterol. In that sequence that I showed you for the SIEBP, there was no indication of how cholesterol could be possibly sensed. However, SCAP, when you look at the sequence of the protein shown down here, has a very unique motif here of 5 transmembrane domains that is known as a sterile sensing domain. This arrangement of 5 transmembrane domains within SCAP is able to actually bind cholesterol if the amount of cholesterol inside of the ER rises to a certain concentration. Okay, so this means we are making SIEBP. It is inserted into the ER membrane and it binds to a bona fide sensor of cholesterol. So now we're getting somewhere. Again, here's the highlighted sterile sensing domain within SCAP. So we're still in the ER. So how can we possibly get this transcription factor to go? So under conditions where, I'm sorry, man, we've got these very nice yellow circles here and the projector is not showing them. I hope this will work out okay. So SCAP, in the absence of cholesterol, assumes one certain formation, and this formation that it assumes in the absence of cholesterol allows it to interact with these SEC proteins, particularly SEC24, which are part of the COP2 protein code complex. Now, for those of you who didn't study this a lot, a quick flashback here, ER to Golgi transport. You guys know who this is? Any ideas? Clearly he likes foxes. I didn't know that either. Think about Nobel laureates and cholesterol. So that guy is this guy. Any idea who this is? Randy Shankman, right? Who won the Nobel Prize, our resident Nobel laureate. Come on, guys. Hi. Right? So Randy's discovery was that he cloned all these important SEC proteins that mediate the ER to Golgi forward transport. Right? And this got him the Nobel Prize together with these other guys that we will not talk about as they are different institutions, including Stanford. Okay, but back to our topic here. So what happens is SCAP without cholesterol, right, with its friend SIEBP, binds to SEC4, and this allows it to undergo forward transport to the Golgi. Now that's still not the place where transcription factor can do its transcriptional thing, right? So now we just went from here to here, but inside the Golgi, and again, there's a little animation that you should have been able to see, and I'm sorry this doesn't work. Once you're in the Golgi, again, our complex here is still together, there are two proteases. They are called site 1 and site 2 proteases. They are only in the Golgi, they are not in the ER. So while this complex is in the ER, they will never see the proteases. Low cholesterol binding to SEC24, forward transport into Golgi. Now you see these proteases, they are bound to the membrane of the Golgi, and they cleave twice our friend SIEDP. And they cleave it first here, that separates this regulatory domain away from our transcription factor domain, and then they cleave right in the middle of the second transmembrane domain, and that liberates now the part that looked like a transcription factor, right? So once you make these proteins, they form dimers, they get immediately imported into the nucleus, they bind to the sterile response elements, and they drive the expression of genes that will upregulate the uptake of cholesterol, which involves the LDL receptor, and upregulate the synthesis of cholesterol, which involves HMG-CoA reductase, which we will be talking about in the second part. So again, the basic mechanism is we are sensing the abundance of cholesterol through SCAP. Our transcription factor is always bound to SCAP, right? If there is no cholesterol around, we have forward transfer into the Golgi, and then exposure to two proteases that cleave SIEDP. Now it can get out of the membrane, dimerize into the nucleus, and enhance cholesterol uptake and synthesis. Again, the little yellow circles that you can't see, they should be there. Okay. Point. So now, when you look at the transfer of proteins out of the ER and towards the Golgi, there's something called the mass transport, bulk transport, where you still get some proteins that may not interact with the COP machinery here, that still get transported out of the ER. So the cell wants to make really, really sure that in the presence of cholesterol, our complex of SCAP and SIEDP does not get into these vesicles and that we are not activating these genes. To assure that there is no accidental transfer here, what happens is in the presence of cholesterol, which accumulates in the ER membrane, there's a conformational change in our protein SCAP here, which has the sterile sensing element. And this conformational change allows it to now bind a different protein. So normally it's able to bind to SEC24, but binding to cholesterol here disengages binding with SEC24, and therefore we don't have active recruitment to these vesicles. But we are gaining also binding affinity towards another protein, which is shown down here in yellow, which is called INSYN, which stands for insulin-induced gene, INSYN. INSYN, as you can see here, also has a ton of transmembrane domains, and this acts as an anchor for our complex in the ER. So again, two scenarios. Low cholesterol, we need to make cholesterol, we need to take it up, we need to synthesize it. Our sensor SCAP is in a conformation that can bind SEC24, forward transport into the Golgi, cleavage of the transcription factor, enhanced transcription of the genes for synthesis and uptake. However, in the presence of cholesterol, cholesterol changes the conformation of SCAP. SCAP no longer can bind to SEC24, but it now can bind to INSYN, which anchors the complex in the ER, therefore preventing any chance of forward transport into the Golgi, therefore not allowing the cleavage of SIEDP, and therefore not allowing transcriptional activation. Makes sense? This is the basic mechanism of how cholesterol sensing can regulate the activity of the transcription factor SIEDP, right? The sensing all happens not within SIEDP but SCAP, and the choice is to either bind to INSYN or to bind to SEC24, and that makes all the difference. Interestingly, when you look at this complex of SCAP, SIEDP, and INSYN that is formed when you have high cholesterol or high sterol levels, it's important to point out that INSYNC itself actually is regulated in its transcription, not just by insulin as the name indicates, but also by SIEBP. So when you have no SIEBP in the nucleus, you're also not making any more INSYNC. I explain why this is important. Under the high sterol conditions, we have this complex that I just pointed out, which is stable and which prevents the processing of SIEBP and therefore transcriptional activation. However, under low sterol conditions, this complex loses its affinity for INSYNC, right? And it gains affinity for the protein components, and we have trafficking and towards the Golgi processing, activation of SIEBP, more gene expression. At the same time, we left our little friend INSYNC here behind in the ER. Now this INSYNC, which is no longer bound to the SCAP and SIEBP complex, actually becomes the target of protein degradation. When it's not bound to SCAP, it binds this protein ubiquitination machinery that leads to polyubiquitination, extraction out of the ER, and degradation. That means you have to replace this, right? We have to undergo a cycle where once INSYNC is broken down because SIEBP is in the nucleus, we have to wait now until SIEBP is fully active and starts to synthesize new INSYNC so that it can function again in its role in capturing this complex. Let's look at this again on this slide here, which had a beautiful animation, which again you cannot see. And I'm sorry about that. All right, let's start with INSYNC again. INSYNC, right, here, can bind SIEBP SCAP when cholesterol is what? High or low? High, right? High cholesterol, this forms a complex. INSYNC is stable, SIEBP does not go process, SCAP stays in the ER. Now, if the cholesterol levels start to drop, several things start to happen. This complex starts to move on. INSYNC is degraded through proteasomal degradation. At the same time, the SIEBP complex here moves into the Golgi. Our site 1 and site 2 proteases cleave SIEBP, therefore it's no longer bound to the membranes, it dimerizes, it moves into the nucleus. Now in the nucleus we know now two groups of genes that get activated in the nucleus. One was genes that are involved in cholesterol synthesis and uptake, LDL receptor and HMG-CoA reductase. But there's a second group that includes INSYNC as well. So both of these genes get activated. Now two things have to come together in terms for the cell to say, stop, enough, I want to shut down uptake of cholesterol, I want to shut down synthesis of cholesterol. On one hand side, you need to have enough cholesterol so that our sensor here, the SCAP protein, can sense that there's enough cholesterol present. But the other thing that needs to be there to efficiently trap again this complex needs to be protein synthesis of INSYNC. So both of these have to come together to form a new complex that traps our transcription factor and inactivates it inside of the ER. This is known as conversion inhibition. Why would the cell go through so much trouble for this process to look at both the renewed synthesis of cholesterol as well as the synthesis of proteins? It's not completely clear. One explanation could be that there are processes that actually require accumulation of protein, such as proteins involved in the synthesis of cholesterol. One moment, let me finish this thought. I saw your hand. That we need to allow the cell to accumulate the biosynthetic machinery for synthesis of cholesterol, which includes HMG-CoA reductase, but also many other proteins, even when there is maybe some cholesterol around. And the reason for that is remember, cholesterol can come from two sources for each cell. One can be the uptake of cholesterol from external lipoproteins through the internalization of lipoprotein receptor. The second source is endogenous synthesis of cholesterol. Why would this matter where it comes from, right? You may say, well, if there's enough cholesterol, there's enough cholesterol. End of story. It doesn't matter whether it comes from the outside or inside. But the protein synthesis machinery that makes cholesterol also makes other important components that the cell needs for its function. For example, ubiquinol, an important part of the electron transfer chain in mitochondria, is a branch of product of the cholesterol synthesis machinery. So the cell wants to make sure that it can not only make cholesterol, but also other important components. So if a cell takes up a lot of cholesterol, it still may want to make a little bit of the protein synthesis machinery that can make other things besides cholesterol, but is in the same pathway. Okay? That's, of course, just our way of explaining something that happens. And all I can tell you is that it happens, but that's our best attempt at an explanation. Conversion inhibition. I want you to think about this and learn it and remember it for next week. Question?
Just at the top, when you have it at the ER level, what is it showing when you're pointing to cholesterol or like what is that explaining?
Okay, so the question is, in case you didn't hear it, what do these errors mean, pointing either towards INSYNC or cholesterol? So this is a little bit unfortunate. What they mean here is that in the presence of cholesterol, this complex is bound to INSYNC. In the absence of cholesterol, this wants to make more cholesterol and travels forward. Yeah, this is not great. It's actually a good point. I probably should cut this out. But this is from a review from Brown and Goldstein, so I should get some. All right, let's go on. All right, this was our regulation of the transcription factor, right? ESA, EBP, senses cholesterol through SCAP. SCAP mediates the trafficking. We looked at the effects once it gets into the nucleus, that you have to make both INSYNC and cholesterol to shut down cholesterol production. Now let's talk about a protein that I already mentioned now several times, HMG-CoA reductase. This is the rate-limiting step for cholesterol synthesis, and this is so long, this pathway, that the word cholesterol got cut off down here. But HMG-CoA reductase actually mediates one of the very early steps of cholesterol synthesis from HMG-CoA to mevalonic acid. A lot of these steps here are, of course, already familiar to you, so I assume that I can ask you any of these synthesis steps next week. All right, just kidding. All right, but here, so, take a deep breath, it's okay. But I do want to quickly point out, see these guys here, geranyl pyrophosphate, for example. These are products that actually branch out of the biosynthesis pathway towards isoprenoid synthesis and things like CoPure. So if you shut down HMG-CoA reductase, you not only lose production of cholesterol down here, but also of some of these other things, like things branching off from farnesyl pyrophosphate, and lanosterols as well, which can be used for the synthesis of hormones, for example, right? So that may be our explanation for this conversion regulation, that you not only have to have cholesterol, because maybe you just gulped up a huge LDL particle and the cell is awash in cholesterol, but you still need to give the cell a little bit of time to replenish enzymes in this biosynthetic pathway to be able to synthesize things that you cannot take up from an LDL particle. Okay, but here it is, HMG-CoA reductase. So the enzymatic reactions, of course, involve just these two small molecules which are water soluble. Therefore, you would expect to find this enzyme sitting in the cytoplasm of a cell. That's not the case. This enzyme, just like our transcription factor, sits inside of the ER membrane. And the idea here is that it sits inside of the ER membrane is because the final steps of cholesterol synthesis happen in the ER membrane, and it basically senses the very end product of these 20 and more biosynthetic steps, and not just the end product of its actual small own little reaction. So really important enzyme. Therefore, because it's so important, it's regulated at multiple levels. We will only discuss two in one real brief. One is very simple, and it brings us back to our friend AMP kinase. Remember that we learned that AMP kinase gets activated when AMP levels rise, ATP levels fall, and the basic response of the cell is to switch off anabolic processes and to engage catabolic processes, such as beta oxidation. One of the anabolic processes that gets switched off that we discussed was protein synthesis. Another anabolic process that gets switched off that we discussed was fatty acid synthesis through the phosphorylation and inactivation of ACC, right? Learn that for next week. But this is not all that AMPK can phosphorylate. It also can phosphorylate and inactivate HMG-CoA reductase, therefore directly influencing how much energy is being spent on cholesterol synthesis. So that's a cool regulatory step because it doesn't involve cholesterol sensing. It actually is an overriding control mechanism for a cell to say, you may need cholesterol, but there's just no energy around to invest in all this biosynthetic activity, and therefore you get reversibly phosphorylated and shut down. There's also some regulation of HMG-CoA mRNA translation. We will not be talking about that. The main regulatory mechanism that I do want to discuss with you is through cholesterol. So this protein, as I said before, sits in the ER membrane here. It has many transmembrane domains, and in fact, it contains the same kind of, I'm losing this now, I thought I had this. It contains the same kind of sterile sensing domain that we found in what other sterile sensor that we discussed today? SCAP. So HMG-CoA reductase, unlike SLEBP, has its own sensor built in. So it's both an enzyme and a cholesterol sensor. Let me modify that. It is both an enzyme and a sterile sensor. Because as it turns out, after longer studies that people perform, HMG-CoA reductase is not really responding to the presence of cholesterol, but actually the presence of another sterol called dihydrolanosterol. This dihydrolanosterol is a byproduct of the synthesis of cholesterol. Remember, I pointed out lanosterol, right? And this, you make dihydrolanosterol whenever you accumulate a lot of lanosterol. That is the molecule that binds and changes the conformation of this domain here, of the sterile sensing domain. So what happens, right? So we've already seen one mechanism of regulation, phosphorylation by AMPK, which switches off enzymatic activity. When lanosterol binds to this binding domain here, it also changes conformation, and what it allows HMG-CoA reductase now to do is in fact quite similar to what happens when SCAP binds cholesterol, which it binds now to INSYNC. Remember, that was the same thing that happened when you have high cholesterol levels in the ER with SCAP binding to INSYNC. So HMG-CoA reductase, when there's a lot of lanosterol around, also binds to INSYNC. The reason that INSYNC has all these extras added to it is that it does not bind to free, simple INSYNC. Instead, remember that we discussed if INSYNC is left without the complex of SCAP, it gets targeted for proteasomal degradation, right? There's this whole ubiquitination machinery that gets recruited. Well, HMG-CoA reductase, when it senses that there's a lot of lanosterol around, does not bind to the free INSYNC. It only binds to the INSYNC that already has this protein ubiquitination machinery bound to it. The consequence of that is that now instead of biotinylating and degrading, ubiquitinating and degrading INSYNC, we are ubiquitinating and degrading HMG-CoA reductase. Okay? Now that is interesting because that's exactly what you want to do if the amount of molecules that you synthesize, particularly lanosterol, is high. You want to switch off this enzymatic activity, and in this case what you do is you polyubiquitinate it, and now you have to remove this protein out of the ER membrane and degrade it within proteasomes. And this step involves a triple ATPase here, and again, you don't need to remember the name, but this extraction process. Remember, this guy here, our friend HMG-CoA reductase, has many transmembrane domains. You have to pull on it to get it out of the membrane. This pulling step requires geranyl. So this molecule here, which as you can see is another isoprenoid, is the second molecule that's required and needs to accumulate besides lanosterol for efficient degradation of HMG and CoA reductase. That is a great idea by every cell, because now you're sensing two direct products that are downstream of the reactions that HMG-A-CoA reductase regulates as a rate-limiting enzyme. Remember, and let me just jump back to that slide. We're right here, granulate, right, is one of the products downstream, and then lanosterol, which can be modified by hydroxylation, is another substrate that is way downstream. You need to have both of these, right, to accumulate. If they accumulate, if lanosterol accumulates, that's a sterol that's being sensed by HMG-A-CoA reductase in its sterol sensing domain, which allows it to bind to INSYNC, but INSYNC that's preloaded with a proteasomal degradation machinery. And then complete degradation of this protein as an added safety mechanism also requires the presence of yet another product that is way downstream of what HMG-CoA reductase does. Again, we need to sense two things. Neither of them is cholesterol to ultimately ubiquitinate and degrade HMG-A-CoA reductase. You may ask yourself, well, why aren't we just sensing cholesterol just like SCAP? It would make things a lot easier for us. No, the reason for that is, again, that you can have two sources of cholesterol. You can have uptake of cholesterol from the circulation, or you can have biosynthetic activity to make new cholesterol. And again, these molecules here, such as lanosterol and granulate, are important to the cell for other biological functions, not just cholesterol synthesis. So you do not want to switch off this machinery just because you took up a lot of outside cholesterol from an LDL particle, because you still need to make things like CoQ, for example, from this molecule here, right? So instead of sensing how much cholesterol is around, you're sensing for this rate-limiting enzyme up here only two steps of the products, but not cholesterol itself, because these are at the level where you can branch off for other biosynthetic pathways. Makes sense? Good. Let's go back. Now, I understand that this is confusing. So we have basically the same kind of response, which is sensing of sterols, if we just want to generalize, right? Causing a conformational change and causing binding to INSYNC. But in the case of HMG-A-CoA reductase, this leads to its degradation and inactivation of function. So you end up with less cholesterol. On the other hand side, when we have the sensing of sterols by SCAP, this leads to an entrapment of SI-EBP in the ER, and therefore, right, the prevention of transcriptional activity and cholesterol synthesis. So the same thing seems to be giving you very different kinds of readouts. So the similarity here is, again, that we are binding at high sterol levels. One binds to cholesterol, one binds to hydroxylanosterol. You trigger INSYNC binding. That's true for both of these. But you are binding to different complexes. Our SCAP and SI-EBP under high sterol conditions bind to free INSYNC and stabilize INSYNC. This complex does not get ubiquitinated, it does not get degraded, it stays stable. Under low sterol conditions, as we discussed, SCAP and SI-EBP move away from INSYNC. This recruits the protein degradation machinery for INSYNC and causes degradation of INSYNC, which then needs to be replenished together with cholesterol when SI-EBP is in the nucleus actively transcribing genes. You accumulate INSYNC, you accumulate cholesterol, you go back to this scenario inactivation. Now, what happens with HMG-CoA reductase? Under high sterol conditions, this time we're sensing lanosterol, hydroxylanosterol, it also gets activated for binding to INSYNC, but it does not bind to the free INSYNC. It binds to the INSYNC that has this ubiquitination machinery already associated with it. And this binding now causes it to be ubiquitinated itself instead of INSYNC. This, in the presence of additional signals, particularly the presence of isoprenoids, allows for the extraction of HMG-CoA reductase out of the ER membrane and degradation, and therefore leads to decreased synthesis of cholesterol. Okay, so makes sense. Let's go through the whole thing one more time. So you see there aren't that many slides today, they are just a little bit dense. All right, we discussed today two signaling mechanisms that respond to the presence of sterols and particularly cholesterol as it relates to synthesis and uptake. The first one was a transcription factor called SIEBP, which can activate the transcription of many genes in the genome that have sterol regulatory elements in their promoters. This includes the LDL receptor, which is rate limiting for the uptake of exogenous cholesterol from the circulation, and it contains HMG-CoA reductase, the rate limiting enzyme for the de novo synthesis of cholesterol inside of cells. So both uptake and synthesis are being regulated by SIEBP. SIEBP itself is synthesized with two transmembrane domains and therefore cannot act as a transcription factor. It's inserted into the ER membrane and it constitutively binds to its friend SCAP. SCAP is the molecule that has the cholesterol sensing domain, the sterol sensing domain in it. This determines whether SIEBP should be active or not. And it determines whether it should be active or not by regulating its proteasomal processing. By regulating its processing through proteases. Let's say proteases. Sorry, didn't want to confuse you. Again, let me say this again. SIEBP binds to SCAP. SCAP has the sterol sensing element, and it regulates the activity of SIEBP by regulating whether SIEBP gets cleaved by proteases, and only the cleavage by these proteases allows it to act as a transcription factor and to move into the nucleus. The sequence of events that we discussed was, in the presence of cholesterol, SCAP undergoes a conformational change in its sterol sensing activity and drags SIEBP into a complex with INSYN. INSYN is an anchor inside of the ER that allows it to trap these two proteins inside of the ER and prevents the forward transport out of the ER into the Golgi. If cholesterol levels fall, SCAP undergoes a conformational change that abrogates binding to INSYN. Instead, it allows it to bind to COP2 proteins and therefore actively facilitates the export out of the ER into the Golgi. Once you're inside the Golgi, SIEBP now gets exposed to two proteases, site 1 and site 2 protease, that cleave in two different locations, liberating the transcriptional factor portion of SIEBP, which forms a dimer, gets imported into the nucleus, drives the expression of many genes. INSYN in this scenario gets left behind. It gets recruited to proteins that mediate the ubiquitination of this protein and its enzymatic degradation. INSYN gets basically wiped out in the ER. So while our transcription factor is in the nucleus, the negative regulator gets diminished. In the nucleus, we are making more HMG-CoA reductase, more LDL receptor, but we're also making more INSYN. So ultimately, now this whole thing here gets reset. We are starting to accumulate more cholesterol in the ER membrane, and we are starting to make more INSYN. So therefore, through conversion inhibition, we can trap the transcription factor again, and that prevents more synthesis of LDL and HMG-CoA reductase. That was one level of regulation, both of uptake and of synthesis. So HMG-CoA reductase is switched on and off in its transcription through activity of SIEBP. But it's in itself also actively regulated by the presence of sterols, as we've seen, and regulated by energy status. I think I'm just skipping ahead of this text here, but you all can read this at a later time. Right? So HMG-CoA reductase. HMG-CoA reductase has the sensor for sterols built into the enzyme itself. Right? Again, a sterol sensing domain. It itself mediates a reaction that really does not generate a sterol, right? It's just at the very beginning of a long cascade of synthesis steps that ultimately lead to the biosynthesis of both cholesterol as well as other important molecules, including isoprenoids and other steroids such as lanosterol. The sensing that happens within the HMG-CoA reductase steroid sensing domain actually is driven by dihydroxylanosterol. This causes it to undergo a transformational change. So if a lot of its end product, or one of its products accumulates, lanosterol, it undergoes a conformational change and it too binds to NSYNC. But unlike the SCAP SREBP complex, it does not bind to free NSYNC. It binds to NSYNC that is preloaded with a ubiquitination machinery that will cause its own polyubiquitination. To completely extract it out of the membranes of CER, you need another product of HMG-CoA reductase activity that is downstream, not lanosterol, but isoprenoids, right? This farnesylate that I showed you. Once both conditions are met, that you have high lanosterol and high isoprenoids, NSYNC gets pulled out and degraded inside of the proteasome, and therefore you need to have new protein being synthesized in order to replenish it. And of course, one way to sense that you need more HMG-CoA reductase protein is through the transcription factors that we just discussed. So these two processes are intimately interlinked. Both of them utilize binding to NSYNC, but two different means. One uses NSYNC to anchor it inside of CER and to prevent the cleavage of SREBP. The other process leads to active proteasomal degradation of HMG-CoA reductase. And of course, the other connection is that SREBP directly regulates how much HMG-CoA reductase protein is being synthesized by driving its transcription of its gene through the SRE that's inside of HMG-CoA reductase. So this is cholesterol sensing in the cell, interaction of two pathways that we discussed now, a transcription factor and an enzyme. I hope that this made sense to you. We have a little bit of time to ask questions. I hope you have some from this lecture. If not, I'm happy to answer some from prior lectures as well. Yes?
For the layer, the SCAP and they move to the Golgi, right?
Yes. Okay, so this was again a question about the movement. So the control of the transcription factor, our nutrient sensing readout here, is really the readout of whether the transcription factor stays in the ER, which is what happens under low sterile conditions, or whether it moves on and gets transported into the Golgi, which happens if there are low sterile conditions. That is really the controlled step that drives this whole process. The sensor for it is SCAP, and SCAP switches between NSYNC binding under low cholesterol conditions and trapping of the inactive complex inside of the ER, or under low cholesterol conditions, it binds to the COP2 vesicles, transfers the SCAP SREBP complex into the Golgi, allows the liberation of SREBP from the membranes and its import into the nucleus, and therefore the biosynthesis of both uptake and generation of cholesterol de novo. Yes?
Can you just review in the degradation of the HMG-CoA reductase how general
Okay, good point. I know this tends to be a confusing question. So the question was, we, as I mentioned, need two things to come together in terms of products of HMG-CoA reductase accumulating. One was this dihydroxylanosterol, which is sensed by the steroid sensing element, and then this allows binding to NSYNC, which is preloaded with ubiquitination machinery. But I also mentioned that you need to have another molecule called granulate to be present for the extraction and active degradation of the HMG-CoA complex. This molecule is required for the activity of a protein that we didn't discuss in much detail that is involved in unwinding, denaturing, and pulling out of HMG-CoA reductase out of the membrane. What that protein is, I'm happy to discuss during office hours, but it's beyond the scope of this class. The important thing to remember is that there are two things that are part of the way downstream biochemical reactions that get initiated by HMG-CoA reductase that are being sensed. One was isoprenols, the other one lanosterol, right? So you need to have accumulation of both of these as a fail-safe mechanism, because both of these molecular groups are actually required for cell function. Besides cholesterol, there are important things that need to be synthesized, such as ubiquinol. Okay, I think there was a second question, but then, yes.
Can you go over why HMG-CoA reductase, like the regulation is so high up in that, like the string there, like you have like the flow chart?
Okay, so the question is, why are we regulating HMG-CoA reductase if it's so far removed from the end products that we're really sensing? That's a great question, and again, we can only guess why nature, made it this way. But the biosynthesis of cholesterol and many of these other products is extremely energy consuming. So if you do not want to make cholesterol, if you do not want to make lanosterol, you do not want to spend a lot of ATP making all the precursors that would otherwise accumulate. So from that standpoint, it would make sense. Okay, time for one more question. Yes?
So is the second pathway when you're doing the HMG-CoA reductase, it doesn't go to the OPS,
Does everything stay within the ER membrane when you regulate HMG-CoA reductase? Yes. This never travels out of the ER. Okay, if you have more questions, I'm actually going to have an extra office hour right after this. So if you want to come with me.